Good morning. This is video number 8, and the topic for today is in video 7 we looked at organelles. In video number 8 we're going to start looking at cell membranes. So we'll look at cell membranes, we'll look at types of passive transport, simple diffusion, osmosis, facilitated diffusion, things like that. We'll look at active transport, pumps, co-transport, counter-transport, and then bulk active transport, the different types of endocytosis and exocytosis. We'll also introduce the idea of receptors. The next video will focus on how receptors work in more detail, but we'll kind of introduce the idea here. We'll look at anchor proteins, and then also we'll look at cell identification proteins as well. So I'll need to add one of those in there. A glycoprotein. There we go. So we'll start off by looking at our free response. The free response will be in a slightly different order than the file I gave you. Again, go to Canvas, go to Files. There's a file that says AP Bio FR referenced in YouTube. And so if you're scrolling through our file, the next one is 2010 number 1. That'll actually be better after the next video, after video 9. So you might go to 2017 number 8 after video 8. All three of these kind of relate to this topic, but you're best prepared for 2017 number 8 after this video. Okay, so to start off with, we want to look at this. So if you think back to video number 1, we were looking at polarity, polar and nonpolar. Water was our example of a polar molecule. Then we looked at long chains of C's and H's, hydrocarbons. In the fatty acid tails of lipids is an example of something nonpolar. Again, lipids are nonpolar because C's and H's have very similar electronegativity. So if I've just got big long chains of C's bonded with C's, obviously a C and a C have exact electronegativity, and so they'll be nonpolar, but the CH bonds are also nonpolar. So one of the things that we saw in our first video was carbon always forms 4 bonds, hydrogens always form 1. So if I want to do kind of a shortcut drawing of, methane, ethane, propane, butane here, then I could draw all these C's with the H's, and I can do a shorthand version like that. At every 4-way intersection, I have to have a carbon. At every dead end, every single bond, I have to have hydrogens. So that is that in shorthand. We then saw an example of a triglyceride. That little rectangle right there is glycerol. And then we bond 1, 2, 3 tails to it. 2 of those tails were saturated with with hydrogens, and this tail was unsaturated. When I add double bonds, cis double bonds, I end up getting kinks in there. So that leads us to today's topic of phospholipids. So you've heard of phospholipids in your class, of course, and your teacher would draw it something like this maybe, and they would say you have a polar phosphate head. Phosphate molecules on their own have 3 negative charges. If I bond them to something, then it has 2 negative charges. If I bond it to something else, it has 1 negative charge. We referenced that a few videos back when we talked about ATP. So basically what we do is we take a triglyceride and we lop off one of the tails. Well, if this was stable with that bond right there, with that big long tail, then if we cut it off, we need to bond something else there. So essentially if this was stable and I break off a covalent bond, I now have to replace it with another covalent bond. So what I do is I replace it on the other side with a phosphate. Because it picks up a bond, it's got a couple negative charges. And so now I have a phospholipid. It'll have 1, 2 tails, and so that's why your teacher always draws phospholipids like this. The majority, maybe all, but the majority, actually I'm going to back off of that. The majority of phospholipids tend to have 1 straight tail and 1 kinked tail. So the question to ask is, if I had 2 straight tails versus 2 kinked tails, what physical properties would that cause that would be different? Well, the more straight tails I have, we remember from saturated fats, they tended to be more solid. When I start putting in kinks, they tend to be more liquid. So our phospholipid here, when we start putting in kinks, this is fairly liquidy. And again, phosphate head, and then non-polar lipid tails. So you might hear the word, it's probably not too important on the AP Bio test, but the word amphipathic Amphipathic means you're both polar, the phosphate head, and nonpolar, the lipid tails. Your 9th grade teacher emphasized this a lot. But you might not have really ever understood why we emphasize this so much in 9th grade. So why do we care so much that phospholipids are amphipathic, that they are both polar and nonpolar? That brings us to this video. And this picture. So in this picture, if I've got a cup of phospholipids and I've got a beaker of water and I dump them on there, the phosphate heads are polar, so they will bury themselves in the polar water. The negative charges on the phosphates are attracted to the slight positives of the hydrogens. By the way, there's more than one type of phospholipid. It's not quite this simple. Other things can be attached on that phosphate head. So if you came across words like sphingolipids or something like that, there's a bunch of different types of phospholipids. The thing that they all have in common is they do have a phosphate head, but they might have other things attached to it. And so that's why there are different types of phospholipids. But at a base, they're phospholipids. All the tails are sticking up out of the water because they're hydrophobic. They're nonpolar, and polar and nonpolar things don't mix. Just like how regular oil, because these are unsaturated, so they'll be oily. Just like regular cooking oil, vegetable oil, if I pour it in water, it won't mix. And if it's less dense, it floats on the top. If it were more dense, it would sink to the bottom and not mix very well with the water. So I get a monolayer of phospholipids. Well then, if I go back to cup of phospholipids and I dump it on there, now I get another layer. And with these, the tails will point down because those phosphate heads, again, I'm making it sound like they have personality, so I'll say they don't like, but what I really mean is they're not stable. They are not stable touching those tails, so they'll always flip around. So because of that, I have a phospholipid bilayer, 2 layers. The inner layer and the outer layer, they call them leaflets. So I have the inner and the outer leaflet. You can have different phospholipids on the outer leaflet compared to the inner. That's why I drew 2 different colors, green and purple. Well now, if I take that phospholipid bilayer, pretend that I have here's my internal leaflet, my inner leaflet. So here's phospholipids, it's floating on the surface of water. Here's the surface of the water, and I'm in the water. And so here's this inner leaflet. Here's the outer leaflet, so I just formed a phospholipid bilayer. Okay, everything is stable. Now, here's the water level floating on top. If I dunk it under the water, then this becomes unstable. Which part is unstable? Well, if I were to look from underneath, everything I would see would be phosphate heads, right? If I look from above, everything I would see is phosphate heads, but on the edge, I would see phosphate, then lipid, then phosphate. I'd basically see something that looks like this. So the problem is, along the edges, the lipid tails from above, most of the lipid tails are buried in there. Until I pull this back, I can't see it. I gave the analogy of a peanut butter sandwich. Bread, bread. If I pull it back, I can see all the peanut butter in the middle. Well, here on the edge, I can see the peanut butter in the middle. Those are the lipid tails. Those lipid tails are now underneath the water. So those lipid tails are touching water. That's not stable. So how can I make sure that these lipid tails don't touch water? Well, if I get rid of edges, then I won't. So if I took my phospholipid bilayer and folded it up into a sphere, I am aware this is a cylinder, but if I could fold it up into a sphere, then basically none of the lipid tails would be touching water. And so whenever I dunk phospholipid bilayer underwater, it automatically makes a sphere. Okay, well, if my hand was on the surface of water like this, and then so here was on the surface of water, and then I go underwater, and I fold it up, then now I'm surrounded by water, but I've also got a handful of water. So what would be in the middle of that sphere? Water. What would be surrounding it? Water. But that's okay, because as you can see, phosphate heads are touching the water. On the outside, purple, phosphate heads touching the water. Everybody's stable. Those lipid tails are buried in the middle. So back to my PBJ analogy, if I could somehow take that sandwich and fold it into a sphere, there are no edges, so there's no peanut butter touching the air or touching the water. Okay, so put that one off to the side. So that's why cells form themselves. Now, eventually, probably won't be in this video, but we'll look at why cells are really small. They emphasize this a lot on the AP-Bio test. If you go back and look at old tests, surface area to volume ratio being really high on small cells and decreasing as cells get bigger is super important. So we will come back to this again, either at the end of this video or during the next video, most likely. All right, so here is our cell. And as you can see, it's made out of phospholipid bilayer. I even kept the colors the same. So here's my cell, purple on the outside, on the outer leaflet, green on the inside, inner leaflet. Same thing here. So here's phospholipids. I just did this as a shortcut, the purple and green lines, but it's phospholipid bilayer all the way around. Okay, so we're going to break this down into a few different categories. There's more than one way to categorize this stuff, but to me, this is a logical way. I think it's the most logical way, but it's definitely a logical way. So what you'll notice is from here over, green, 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 and then this doesn't have a protein in it. All of these are passive. So we can kind of put a dividing line like that. Passive. Everything to the left of this dotted line is passive, 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 passive. Everything over here is active to the right. Then we get to this dividing line. This is also active, but it's what we call bulk. So that word, if you can't read it, depending on what you're seeing, this is passive. Everything to the left is passive transport. Everything to the right is active transport. But this is more like individual molecules. Now it might be not one at a time, it might be two at a time or three at a time. You've seen sodium-potassium pumps maybe, moving two or three molecules at a time. Over here is active transport bulk. That's where you're moving a lot. We're moving things in bulk. And then over here, we're just going to call this other. So with transport, you actually have to be moving something in or out when we start calling it passive and active. Over here, when I get to a receptor, you know, we'll see that a ligand might bind to this receptor, and so the message gets inside, but the actual ligand doesn't go inside. So I'm calling everything over here other. So we're going to start out over here. So we'll begin, and I'll write in green since it's passive. We'll begin with simple diffusion. That says simple diffusion. And with simple diffusion, basically, if something is small enough and non-polar enough, it can squeeze through, it can just slide through this phospholipid bilayer. It can literally just squeeze between those phospholipids. Because one of the things that's interesting about phospholipids is this. If you look over here at this picture, this is an actual image, it's not computer generated, of a cell membrane. And if I look at the cell, so the cell is, you know, however big this, if I kind of complete that arc, I can see how big the cell is. So we're zoomed in a lot. That's the cell membrane. And if I look at that, then this is obviously art, not a real thing. It's phospholipid bilayer. So the average phospholipid bilayer, of course it depends on how long the tails are, but the average phospholipid bilayer, if I had a phospholipid bilayer and I stacked another one on top and another one on top and another one, pretend that they wouldn't repel each other or anything, on average, I could stack about 8 to 10,000 of them on top of each other, and that would equal the thickness of a single sheet of paper. So a phospholipid bilayer is, I'll just pick the middle number there in that range. A phospholipid bilayer is maybe 1/9,000th the thickness of a sheet of paper. And then what's interesting is, well, that's interesting too, but what's interesting is that like that phospholipid, that green one, is not bonded to its neighbor. It's also not bonded to the purple one across the way. So in this instance, that means that they're just like floating around. Because they're unsaturated, they're oily. They're not all saturated bonds, right? Remember, one of the bonds tends to be unsaturated and one saturated. So if you think of the pin caps as the phosphate heads, and the rest of the pin as the lipid tails, then this is what a phospholipid bilayer looks like, right? Phosphate heads, lipid tails. Phosphate head, lipid tails. And they can slide past each other because they're not bonded. So like that purple, that blue, that red, that green, they're not bonded with each other. So laterally, they move quite a bit. Now, why is it that this is rare? Why don't they flip-flop? Why doesn't that purple guy from the outer leaflet move over to the inner leaflet? And vice versa. Well, the reason why is because now that phosphate head has to be exposed to the lipid tails. So that doesn't happen very often. But it is kind of weird that all that's holding my cells together, an average human is supposed to be approaching maybe 100 trillion cells. So I'll just say trillions. If you're the bigger you are, the more cells you have. Your cells aren't bigger, you just have more of them. So 350 pounds, 7 foot tall basketball player just has more cells than I do. They don't have bigger cells, just more of them. So what that means is like all that's holding my cells together is 1/9,000th the thickness of a sheet of paper of phospholipids. Those phospholipids are supposed to be, when we don't have proteins in there, they're supposed to be the consistency of salad oil. That's according to my molecular biology, the cell textbook that I reference every once in a while. They compare it to salad oil. So I'm going to call that like kind of like olive oil. All right. And that's all that's holding me together. And each phospholipid isn't bonded to its neighbor. So why doesn't it fall apart? Well, the reason it doesn't fall apart is because if that green guy started to like exit and go towards the middle, there's water, the water pushes it back. If the purple guy starts to like exit, the water will push it back. So really all that's holding your cells together is the fact that the middle part, those lipids, don't want to touch the water on the inside of your cell, and they don't want to touch the water on the outside of your cell. So back to phospholipid bilayer right here. As soon as, again, above me would be water and below me would be water. As soon as that guy starts to kind of like peek out, water will push it back in. Just like if I have oil and water mixed in a beaker and I swirl it, the oil will get pushed back out again, back up to the top, no matter how many times I stir it. So that's what's holding you together. Now that's not all that's in there, because according to simple diffusion, if you're small enough and nonpolar enough, you can just squeeze through. So for example, here's some green dots. I've got a high concentration of green dots on the outside. I have a low concentration on the inside of the cell. So what does that mean? Well, according to simple diffusion, things spread out until they're about equal. So these will spread out until they're about equal. Well, if I've got 2, 4, 6, 8, 10 over here and 2 over here, they're not equal. So these guys are going to be moving back and forth, but the net movement will be these guys moving here. So yeah, these guys could leave, but for every one of these that leaves, there's going to be 5 of these that enters, because we have basically a 1-to-5 ratio, if I counted that right. 2, 4, 6, 8. Yeah, I've got a 1-to-5 ratio. So overall, the net movement will be inside. I'm going to make that a different color so it's easier to see. So these dots will move from high concentration to low concentration. We'll say we have a concentration gradient. If they were equal, then we would not have a concentration gradient. So a concentration gradient is where you have a difference in concentration. And the bigger that difference, the faster things will move in. So they'll move in until they're at about equilibrium. There might be other forces at play. If somebody was positive or negative or whatever, there might be attraction or repulsion, but we'll ignore that for the moment. Things will move from high to low concentration until they reach equilibrium. That's simple diffusion. You might hear it called moving down the concentration gradient, because I'm going from high to low, so I'm moving down or with the concentration gradient. The opposite of that is moving up or against the concentration gradient. So that's simple diffusion. As long as I like green dots, who doesn't like green dots, then that's great. Green dots are going to keep entering. So simple diffusion. Does that take any energy on the part of the cell? No. Yes, I understand when things are moving, that's kinetic energy, but the cell isn't doing it. Just like over here, I've got a window that's open. And if the breeze comes in, the house didn't have to pull that breeze in. The molecules are just diffusing on their own. If there was smoke outside that window from a fire, the smoke particles would just diffuse in on their own and I would smell that there was a fire. Okay, well what if we have something that's maybe polar and it has difficulty getting through, like water? Well, then we can set up a channel. So this is called a channel, and its process is called facilitated diffusion. Facilitate means to help. So what is that thing actually called? A channel. What does it do? Facilitated diffusion, right? What is that thing out there called? That's called a car. But what is its function? Well, it gets me from point A to point B. It's a type of transportation. What's that thing over there? That's an airplane. But what does it do? Well, it flies. So what is that? A channel. What does it do? Facilitated diffusion. Facilitate, F-A-C-I-L-I-T-A-T-E, I think. Facilitated diffusion means helped diffusion. Facilitate means to help. So that channel is helping something get through. So let's say that I've got something polar. Let's say these blue dots are polar. So without a channel, they would just like bounce right off. They can't simply diffuse in, so they need facilitated. They need helped diffusion with a channel. So same thing, high to low, but I just opened up a passageway for them. Now realistically, these proteins aren't just wide open channels to where like anything small enough can get through. They have like their structure. Remember protein structure from video 2 or 3, somewhere in there. I have primary, secondary, tertiary, quaternary structure. So based on the way that they're folded up, they'll have all these like nooks and crannies. That's an old person word. So say here's my channel. They'll have all these little nooks and crannies so that maybe they're really good at only letting in calcium or potassium or something like that. So potassium has an easy time like weaving through this channel based on size and charge and everything else, whereas calcium does not. So this might be a sodium channel, and it's pretty specific for just sodium. I'm not saying that 1 in a billion times something else doesn't get through, but it's really specific for just sodium. One that we've studied quite a bit is called aquaporin. It ends in IN, so it's a protein. It makes a pore, and it lets in aqua, water. So I might let in water with aquaporin channel through facilitated diffusion, and that's passive. It does not require energy on the part of the cell. Over here, I have a gated channel. Gated, it's got a gate on it. So that means the cell can decide when to let something in and when not to let something in. With a regular old channel that's open all the time, anytime these blue dots are around, they're going to move in. Well, now I can decide when to let them in. So I've got a little bit more control. I can't control which way they're going to go. Once the door's open, they will move from high to low. In this scenario, they are going to move in. I could have high on the inside and low on the outside. Then they would move out. But I can at least shut the door when I want. Back to the smoke analogy. If there was smoke outside the window, I can close the window. Now the smoke won't get in. If I open the window, then smoke can get in, but I have no control over which way it moves. It will move in. So that's what's happening with a gated channel. We see these in like on neurons. We have gated, voltage-gated sodium ion channels and voltage-gated potassium ion channels. Also passive transport, that's why it's still in green. Then this over here is, got it right in green, transport. They might just call it a transporter, a transporter. These still follow a concentration gradient, but they kind of change shape. These channels are a little bit more passive. They're more like a rigid sieve. Let me adjust my blinds over here. It's a little bit better. So with this, it's kind of more like just a rigid hole, like a sieve. And this guy has, you can kind of see, hopefully, that there's a specific shape that it can receive. So let's say I've got this kind of rectangle thing right there. That rectangle thing fits in, and when it does, because of chemical interactions, the stable shape of this object changes. So it's kind of like this. Pretend that this is sort of V-shaped like this. So Everything above the V is outside of the cell. Everything below the V is inside the cell. Okay, so here we go. As soon as this brown thing touches, the most stable chemical shape of this protein is like this. But now that it let that thing in, its most stable shape is back to this. So anytime the brown rectangle comes in, it'll let it in, just like that. If something else comes along that's a different shape, like this, when it hits, it doesn't fit properly, so it doesn't get to go inside. However, we're still moving from high to low concentration. So we're still obeying the laws of diffusion. So here, things will move from outside to inside in this example. Okay, that is passive transport. Trying to think if there's anything else I wanted to mention on those. Okay, so now we're going to look at active transport. What if, I don't know, what if I love these brown dots? Yeah, there's 2, 4, 6 on the inside of the cell and 1 on the outside. But what if I want to hoard it, kind of like we're hoarding toilet paper? Sorry for the toilet paper joke. So if I want to hoard it, then now, if I just had an open channel or a transport, if I had any of these, then those brown dots would leave. But I want to pile them up. So sometimes there's a nutrient or something, for example, that we want to pull in as much as we can. So now I have to move from low concentration to high concentration. So in order to get this guy to be pumped in, that says pump, then I might need to burn an ATP. So I'll take an ATP, I'll break off a phosphate, I'll call it an inorganic phosphate. The abbreviation for that is P sub I, P subscript I. I don't know why I think that's confusing. I think you should say PO4, because when you put PI, that makes me think inorganic phosphorus, because P is phosphorus. But that is the official abbreviation. So why do we care if we call it an organic or inorganic phosphate? Doesn't matter in AP Bio. It just, I break off a phosphate, that provides the energy to pull something from low to high concentration. So one of the reasons that I have to eat food is so that my mitochondria can convert food into ATP so that I can run my pumps. They might also call these pumps ATPases. So like, for example, the sodium-potassium pump that I mentioned just a little bit earlier. So it's going to push sodium out of the cell and pull potassium into the cell, and it requires energy. So some books will call it a sodium-potassium pump. Others will call it a sodium-potassium ATPase, letting you know, pump, ATPase. If it's ever called a pump, that means it's running on energy and it's probably ATP. So sodium-potassium ATPase. It is possible to have a pump run on something else. We saw in the cellular respiration videos last week that proton pumps ran from energy provided by electrons falling in energy levels. So you don't have to run on ATP. We also saw, if you did the practice-free response from last week, that ATP and GTP can both be energy sources. We'll talk about GTP a little bit over here later, likely. So it doesn't have to be ATP, but it usually is. So that's a pump or an ATPase. Then over here, now I've got orange dots, high concentration, orange dots low. Purple dots, high concentration, purple dots low. Okay, what if I wanted to get rid of orange dots? So I want to somehow push these orange dots from low concentration to high concentration. I want them out of my cell. So there you go. I want the orange dots to go this way. Well, what's the problem? I'm going from low to high, so that's going to require energy. Let's say I'm not going to use ATP. I want to come up with another clever method. Well, then what I can do is I can take the energy from the purple guys moving. Pretend that purple, I don't care about purple. If they come in, fine. If they leave, fine. I don't really care. So now purple will automatically go from high to low. That can provide the energy to run this protein to push these guys from low to high. So you might remember the example I gave in class. It's going to be tough to give here, but pretend that I'm in a doorway. So here's a doorway around me, and we're looking from above. It's a circular door, like you see at airports or fancy hotels sometimes. And remember, the ideal was, let's say that I'm now in the doorway. So here we are. And again, it rotates this way. So purple automatically moves, I guess, out of the hotel. So we're in the hotel and we're going to move out. Hopefully this analogy makes sense on camera. Made sense in class. So the door goes this way and it'll go out. So the purple is automatically going through the doorway. The orange is not. But if we can just get the orange and the purple into the same door, the purple can kind of push that revolving door for the orange one. Just like me, if I had a 2-year-old, he's up walking around, but he's not strong enough to maybe push that door. So we'll go in, we'll get into the same compartment. I do the pushing, and then he gets to just sort of like tag along. We're going the same way. So we call that process co-transport. Co-transport, we're going the same way. Well, if I look at this one, let's say I want some brown dots. I've already got quite a few inside. I want them inside. There's one out there, so I want the brown dots to move this way. That's fine. Maybe I don't care about the green dots. So now I let the green dots passively move out, and that provides the energy to pull this in. The analogy there would be, let's say that here we are. I'm inside the hotel. I'm a terrible parent, so my 3-year-old was outside. Wait, he was 2. We've been at the hotel for like a year, I guess. So my 2-year-old is back over here. He can't get in because he can't push the door. So I push the door and then he can just walk in. I'm providing the power of pushing the door and then he just moves in on his own. Or he's actually like shoved in. The door's actually like pull it. Once he's in that little triangle cutout, he's actually getting pulled in. So they're going opposite ways. So we would call that counter. Again, I know that might be hard to read, but that says counter transport. So co-transport, we're going the same way. Counter is opposite. You know, you have like point, counterpoint, or attack, counterattack. It's the opposite. Now, you might be wondering, how are these classified? Like, is that active? Because, I don't know, the cell's kind of harnessing energy from here to go to there. So is it active? It's still not really the cell spending energy. It's classified as active. You can argue about whether or not you agree, but science says we're calling it active. So all these green ones, passive. Brown, 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 active. But all of these active methods that we're using right here, one of the things you'll notice is they're bringing in like individual molecules, one at a time, maybe 2, maybe 3, maybe 4. So small amounts of molecules, if not individual. What about bulk? What if I need purple dots and I need 50,000 molecules of whatever the purple dot is, and I need it like now? Well, I could be bringing stuff in through channels or pumps, 1 or 2 at a time, and that'll be really accurate. So now when I bring in 10,000 purple dots, I bring in 10,000 purple dots and probably nothing else. Or maybe I make one mistake and I somehow get 1 molecule of a non-purple dot in there. So I'll be highly accurate, 99.99 out of 10,000. Yeah, 99.99 out of 10,000. Correct. Super accurate, but kind of slow. Just like if I'm letting students into the building. If I check every individual student's ID as they come in, and I let them in 1 at a time, I'm probably not going to make many mistakes. But what if I need to let in 500 students in like 15 seconds? Now I'm just checking some IDs. So it'll be fast, but less accurate. That's what bulk transport is. Let's say that I want these purple dots. Well, right outside the cell, there's purple dots, red dots, and green dots. Sorry if you're colorblind. And so what will happen is the cell membrane, the phospholipid bilayer, will just kind of like pinch inward, almost like you're blowing a bubble, except you're sucking it in. So earlier in the year, apparently not many students had done this, but you can take your bubble gum, kind of like flatten it on the front of your teeth, and then suck in. That's what we're doing. So we're bringing something into the cell. So this is a type of endocytosis. So endocytosis. Osis is a state of or a condition of. That's why some medical things are in osis, like scoliosis. Oh, you have a condition of. So it's a condition of site, cell. We saw that word earlier, or that word part earlier, and then endo, inside, like endothermic. So it's a condition where we're bringing in something into the cell. Well, when I do that, if I just kind of like pinch inward, like here's my cell membrane, and I just kind of go like, then yeah, I might have gotten some purple dots, but I also got red and green and everything else. So I got some of these purple dots that I wanted, and it was quick, but it was less accurate because I also got red dots and green dots. And everybody knows you don't want red dots and green dots. I mean, come on. Okay, we've got two types of endocytosis. Phagocytosis. That's if you're generally bringing in solid particles. Phago means to eat. And then pinocytosis. That's when you're bringing in dissolved particles. That's more like drinking. So just like if I said I was eating a hamburger, we call that eating. And then if I'm bringing in liquids, I call that drinking. So that's what those mean. So if I'm bringing in dissolved particles and it's more liquidy, pinocytosis. If I was like a white blood cell gobbling up an E. coli or something, that would be phagocytosis. Okay, next step. So why have multiple ways to bring stuff in? Why not just bring it in this way, or this way, or this way? That's super accurate, but it's slow. This over here is faster, but it's less accurate. So sometimes you need to be fast, but less accurate's okay. Other times you have to be super, super accurate. Well, now we have a medium one. This is called receptor mediated endocytosis. So receptor mediated endocytosis. And receptor mediated endocytosis is really cool looking. So like right here, to me, I can't see like anything that looks super solid. So you can see the cell membrane is kind of pinching in right here, like we see up here. So I would say like, all right, they're telling me it's going in and not going out with the arrows. So I'm like, that's a type of endocytosis. I'm going to call it pino because it looks like I don't have large chunks. Here, I know he's yellow, but this yellow guy is a white blood cell and he's gobbling up an E. coli. So he's bringing in solid particles. That's phago. Then we have receptor mediated endocytosis. Receptor mediated would be this. I have these receptors up here that are complementary to triangles. I've got the green triangles up here. So now what I do is I basically send a bunch of receptors to the surface of my cell. So now instead of just grabbing what's ever right outside the cell, here's my cell, like that. Instead of just grabbing whatever is right outside the cell, I have a bunch of receptors that will grab green triangles. Now whenever I pinch inward, I'm still going to grab other crap too, but I'm also getting more of the green triangles. So these green triangles will stick to the receptors. And then as the cell kind of pinches inward, now I have more guarantee. I don't really have a guarantee. If there's no green triangles out there, I can't grab them in. If there's only two, the most I can grab is two. But if there's quite a few, then I'll probably fill up most of these receptors. And as I pinch inward, now I'm going to have a good amount of the stuff that I want, green triangles, and I'll have less of the stuff I don't. Once again, though, now we need to send the receptors up to the surface of the cell, pause for a moment so those receptors can fill up, and then pinch inward. So because of that, it's medium accuracy, medium speed. So these things over here, individual, maybe 2-3 molecules at a time, super accurate but slow. Pinocytosis, that's super fast but not very accurate. And then receptor mediated endocytosis, that's kind of like medium accuracy, medium speed. So now your cells can adjust and do what they need to do, just like homework. If I need to get a 10 on this math assignment to get the grade that I want for the year, I'm going to finish it, but I'll probably be pretty sloppy. If I know that I need like a 98 on this math assignment to get the grade I want for a year, I'm probably going to take my time and focus on accuracy. We can also ship stuff out of the cell. We call that exocytosis for obvious reasons. By the way, here's what it looks like when we're pulling stuff in. You can kind of see we sort of make this cage-like structure out of these molecules called clathrin. That's beyond the AP Bio test, but it would show up in like a cell biology class. The molecules of clathrin are kind of Y-shaped. All the Y's kind of line up and help like pinch into like suck in something from a cell. Here's receptor mediated endocytosis. You can see as it pinches in, the inside layer is coated with all the stuff that we want. In my picture, it was the green triangles. All right, next up, we can also do exocytosis, which is just the reverse. I might, let's say this is a gland and it's secreting some sort of hormone. Well, it needs to like barf it out. And so that's exocytosis, the condition of exiting the cell. So now I might have something like this. I manufacture green dots, I make a vesicle of it, and then that phospholipid bilayer goes out and then just like that. Because when phospholipid bilayer bumps into phospholipid bilayer, two become one. In a previous video, we mentioned soap bubbles. When one bumps into another, they sometimes just combine to become one. I might, let's say that I suck in this bacterium, phagocytosis. I surround that vesicle, they call it an endosome at that point. No worries, just wanted to be accurate. I surround that endosome that I just brought in. You would be calling it a vesicle probably. I surround it with lysosomes. The lysosomes then destroy the bacteria. Then I could do exocytosis and barf out all the bits. So here's my green bacteria. I might do phagocytosis to bring them in, chew them up into a bunch of pieces, chemically digest them, and then I do exocytosis to barf out the bits. All right, so passive and active transport. The next thing that we wanted to look at is this portion over here, receptors, enzymes, or receptor linked to an enzyme. That's what we call a G-linked protein, anchoring proteins, and cell identification proteins. But according to my timer, I'm between 40 and 45 minutes, so that's where we're going to stop at the moment. We'll come back and get this in the next video. Thanks.